Okay, so during the previous presentation, I've already discussed that we have two types of nuclear division, namely the mitosis wherein the nucleus divides and the meiosis wherein it is a reductional cell division, wherein it produces daughter cells that are half or contains half the genetic information of the parent cell. And we also have the cytokinesis wherein the cytoplasm divides and the cytoplasm of the cell is splits into two, making two new cells. So cytokinesis begins at the end of mitosis and meiosis. Today, I am going to discuss the phases and substages of meiosis 1 and meiosis 2. Meiosis preceded by interphase, which include the chromosome replication. It is consists of two meiotic division, namely the meiosis 1 and the meiosis 2. Meiosis is also called as the reductional cell division. The original cell of the meiosis is diploid or 2N. And at the end of meiosis 2, 4 daughter cells are produced that are haploid or 1N. It also produces gametes such as the egg and sperm cell. It occurs in the testes in male for the production of sperm cell or what we call the spermatogenesis, and in the ovaries of female for the production of ovum or egg or what we call the oogenesis. Okay, the importance of meiosis are it ensures all organisms produced via sexual reproduction contain the correct number of chromosomes. And lastly, meiosis produces genetic variation by the way of recombination. Okay, mitosis and meiosis is composed of same phases which include the interphase. So as we all know, interphase is the longest phase in the cell cycle, and during the interphase, the cell prepare for the cell division, and the cell will continue to take nutrients and keeps on growing. Interphase of meiosis 1 is also composed of 3 phases, namely the gap 1 or G1 phase, wherein it is characterized by the growth and development of the cell, followed by the S phase or synthesis phase, wherein cell actively duplicates its genetic information in the preparation for the next cell division. And lastly, we have the gap 2 or the G2 phase, wherein there is a rapid growth of the cell and protein synthesis continues in preparation for mitosis or meiosis. Followed by the interphase is we have the prophase 1. Okay, during the prophase 1, the homologous chromosomes paired and exchanged DNA to form recombinant chromosomes. The prophase 1 is divided into 5 phases, namely the liptotin, zygotin, pachytin, diplotin, and diakinesis. For the liptotin of prophase 1, during this phase, the chromosomes start to condense. In addition, the appearance of the chromosomes at this stage of prophase 1 is likened to a string with a beads, and these beads is what we call the chromomere or the centromere. Next is we have the zygotin. During the zygotin, homologous chromosomes become closely associated to form pairs of bivalent consisting four chromatids or tetrads. Okay, so when we say synapsis, it is the pairing of homologous chromosome or the bivalent. In addition, synapsis can form either in the lower or upper portion or part of the chromosome wherein it creates contact called the synaptonemal complex. So this complex can be compared to a zipper-like structure. And lastly, this complex also facilitates the synapses by holding the chromosomes together. Followed by the zygote is the packeting. During the packeting, there is crossing over between the pairs of homologous chromosome or the bivalent. Okay, so when we say there is a crossing over, there is also a exchange of chromosome between the exchange of chromosome segment between the two chromosomes. During the phase or stage of diplotin, the homologous chromosomes start to separate but remain attached by the chiasmata. When we say chiasmata, it is the point at which the chromosome remain attached and at which crossing over and exchange of genetic materials occur between the two strands of the chromosome. And lastly, we have the diakinesis, wherein homologous chromosomes continue to separate and ready for metaphase 1. During the metaphase 1, the homologous pairs of the chromosome, or what we call the bivalents, arrange as a double row along the metaphase plate. In addition, the two pairs of centrioles align to the opposite poles of the cell. During the anaphase 1, the homologous chromosomes in each bivalence are separated and are ready to move to the opposite poles. And for the last phase or stage of the meiosis 1, we have the telophase. During telophase, the chromosomes become diffuse and the chromosomes gathered at the opposite ends of the cell. And lastly, there is a new nuclear membrane that reforms or around the cluster of each new cell. After telophase 1, cytokinesis will take over. It is the final cellular division to form new cells followed by meiosis 2. Lastly, meiosis 1 is a reductional cell division wherein the original diploid cells had two copies of each chromosome and the newly formed haploid cells have only one copy of each chromosomes. For the meiosis 2, it separates each chromosomes into two chromatids, and the event of meiosis 2 are analogous or the same to the mitotic division or mitosis, although the number of the chromosomes involved had been divided or half. Okay, take note that there is no interphase in meiosis 2. Kasi after ng cytokinesis sa meiosis 1 natin, the cell will continue to divide or the process will continue into meiosis 2 without the interphase. In prophase 2, there are two daughter cells with each with 23 chromosomes or 23 pairs of chromatids. In each of the two daughter cells, the chromosomes condense again into a egg-shaped structure. There is also a plasma membrane or the plasma membrane dissolved, the centrioles duplicate, and lastly, the spindle fiber will form again. For the metaphase 2, the chromosomes line up in the center or in the metaphase plate. And lastly, the spindle fiber attached to each sister chromatids. In anaphase 2, the sister chromatids are then pulled to the opposite poles due to the action of spindle fiber. And lastly, the separated chromatids are now individual chromosomes. In telophase 2, the chromosomes completely reach the opposite poles. And lastly, there is a new membranes that forms around each set of chromosomes to create two new cell nuclei. Telophase 2 is the last phase of meiosis 2. However, cell division is not complete without another round of cytokinesis. During the cytokinesis, there are four granddaughter cells, each with half set of chromosomes. In male, these four cells are all sperm cells, and in female, one of the four cells is an egg cell, while the other three are the polar bodies. To sum it up, meiosis has 8 stages in total, plus the interphase in meiosis 1. We have the prophase 1, metaphase 2, anaphase 1, metaphase 1, anaphase 1, telophase 1. We have the prophase 2, metaphase 2, anaphase 2, and lastly, we have the telophase 2. Meiosis occurs in germ cell, in egg and sperm cell only. It produces 4 haploid daughter cells, and the chromosome number is half in each daughter cells. There is also a genetic variation, or yung genetic variation na yun nangyayari or it occurs in the process of crossing over. So when there is a crossing over kasi dun sa may stage ng prophase, substages ng prophase 1 natin kanina, diba? The chromatids are swap their genetic materials to the other chromosome. That's why they are no longer identical to the parent chromosome. Kaya nga nagiging unique tayo or tayo as individual. We are unique because of the crossing over. Kasi kung walang genetic variation, i-carry out natin or meron tayong the same genes as the parent, same sa parent natin. So, ibig sabihin, hindi na magkakaroon ng recombinant chromosome. And lastly, the daughter cells are distinct from the parent. Yun nga, na-mention ko kanina because of crossing over. The importance of meiosis are ensures all organisms produced via sexual reproduction contains correct number of chromosomes, and also produces genetic variation by the way of recombination. And lastly, it produces gametes such as the sperm and egg cell.